Welcome to this very first webinar hosted by the Recomb project. It's exciting to see so many of you here. In this webinar, we will not only shed light on the status and progress of this important research project, but we will also discuss gene therapy and how this works for RAG-SCID patients. Thank you all for taking the time to join this session. It's a full program, so let's begin. My name is Johan Prevot. I'm the Executive Director of IPOPI, the International Patient Organization for Primary Immunodeficiencies. And it's my great pleasure to moderate today's webinar. I would like to take a moment to thank the European Union's Horizon 2020 Research and Innovation Program for its invaluable support towards the Recomb project. Joining us today for the webinar are three expert speakers: Professor Arjan Lankester, Professor Karin Pike-Overzet, and Professor Frank Staal from Leiden University Medical Center in the Netherlands. Welcome to you all, and thank you so much for joining us today. Before we start, I would like to give you a quick overview of the different parts of this webinar. The first presentation by Professor Lankester will introduce SCID, the current treatment approach with allogeneic stem cell transplantation, and the reasons to develop gene therapy as an alternative curative treatment. Once the presentations begin, you can pose your questions in the question tab that you see on the right side of your webinar screen. The questions will then be brought up for discussion after all three presentations have been held. So with these explanations done, let's get started. It's my pleasure to welcome our first speaker of the day, Professor Arjan Lankester. Professor Lankester obtained his medical degree at Leiden University Medical Center and his PhD in immunology at the University of Amsterdam. He was appointed as Professor of Pediatrics and Stem Cell Transplantation at the University of Leiden in 2016. Professor Lankester, the floor, if I may say so, is yours.
Thank you. Thank you so much, Johan, for this very nice introduction. Dear participants, it's a great pleasure to present to you, together with my two colleagues this afternoon, where we think we stand at this moment with the treatment of patients with RAG-1 deficient SCID. And what we will try to do is to take you through the developmental course that we are currently going through, and hopefully give you an up-to-date information with our presentations. So if we go to the first slide, yes, I'm in control. So let's first talk about what we're talking about when we mention the name SCID. So SCID stands for severe combined immune deficiency, and it is typically characterized by the absence of T cells because of a lack in T cell development. And this lack in T cell development may be associated with the absence of B and NK cells as well, but not necessarily. It's depending on the genetic defect. It's a very rare disease, and it occurs in about 1 in 50,000 newborns, a little bit depending on the ethnic background of the population. SCID patients do not present with a genetic defect or a SCID patient, but they present with clinical symptoms that are mostly life-threatening infections and a failure to thrive. And when untreated, these patients usually die within the first year of life because of these complications. So we are really talking about a severe disease and a medical emergency to deal with. So when we go a little bit into the disease itself, I've shown you here two cartoons. On the upper left, you can see the different types of SCID as we know it today, different because of different genetic defects that all cause more or less a similar clinical phenotype. And if we go to the lower right, you can see a cartoon produced by my colleague Karin Pike some time ago, in which you can see, going from left to right, development of lymphocytes. In the upper curve, the B cells, and in the middle line, the T cells, from its early progenitor cell to, on the full right, the mature cells as we can detect them in blood. And what you can see is that there are different stages, all indicated with arrows and with vertical lines. And all these stages can be, let's say, stopping points in case there is a genetic defect. So as a consequence of a genetic defect, the development of either T cells or T and B cells can be interrupted and resulting in the absence of a mature T and B lymphocytes, causing the disease SCID. And as encircled with a green circle over here, you can see the favorite gene of this afternoon, RAG-1, causing SCID. So what are the challenges when you're confronted with patients with SCID? Well, the first thing is to do a life-saving procedure because of the severity of the disease. And the aim is not only to save the life of these kids, but also to restore as soon as possible protective T cell immunity, because that is the major Achilles heel of this disease. And not only to restore T cell immunity, but if as much as possible, also restore thymic function to ensure a long-lasting reconstitution of the T cell immune system. But in addition, you would also like to have restoration of the antibody production caused by the deficiency in the B cells. And above all, you would not only like to restore this, but you would like to restore this lifelong. And remember, we are talking about neonatal patients, and the procedure should actually be a lifelong acting cure. And doing this, you would like to do this with as least as possible harm to the patient, so preventing acute and long-term toxicity as much as possible. So how do things happen in you and me, assuming that we're all healthy individuals? So what you can see over here in this cartoon, that from the early progenitor cells in the bone marrow and in the thymus, there is a production of mature T cells. This is the situation that is ongoing every day in all of us. In SCID patients, this process is interrupted. So although the stem cells are there, they do not develop into mature T and B cells, causing the clinical disease SCID. So how do we manage this problem nowadays? Patients receive allogeneic stem cell transplantation, and the procedure essentially implicates that healthy donor stem cells from an as much as suitable donor are being administered to the SCID patients to restore the immune defect caused by the genetic defect in the patient itself. So after a successful stem cell transplantation, ideally, you should have a restoration of T cell and B cell immunity. And this procedure is being used already more than half a century with success, of course, also accompanied by an enormous learning curve. So what are today still the challenges when treating patients with allogeneic stem cell transplantation for SCID? Well, to bring the patients to a transplant in the best possible condition to do a safe transplant procedure. But for a transplant procedure, you need a suitable donor. And I'll come back to that in a second. Of course, you would like to ensure optimal and persistent stem cell graft function. And you would like to do this with the least possible harm, and particularly to avoid the typical allogeneic stem cell transplant related complication, graft versus host disease. So let's go to the donors. We usually try to find as much as possible matched donor, matched with respect to tissue typing, HLA typing. And we always start to look in the family whether there is a sibling, brother or sister that has the same HLA type as the affected patient. And in some families, you find such a donor, and that is on paper the ideal stem cell donor. But this happens only in about 20% of the cases. So what to do with the other 80% of the patients? In those patients, you try to find an alternative donor, either a volunteer donor from the International Donor Bank or a mismatched family donor. But of course, this also has its limitations. And quite often, at least in a majority of cases, you will not find a properly matched donor. Does it have implications for outcome? Yes, it has. This is just a graph to show this with help of the RAG-SCID database results. And what you can see in this graph is the overall survival after stem cell transplantation using either a fully matched donor or a mismatched donor. And as you can clearly appreciate, the overall survival when using a mismatched donor is clearly inferior compared to using a matched donor. And I'm only talking about survival over here. Graft versus host disease, the typical complication that can occur in allogeneic stem cell transplantation, even if you use a fully matched donor, really has an impact on the overall survival in SCID patients. So SCID patients that develop significant graft versus host disease have a much less favorable outcome compared to the ones treated with a matched donor. So these are just two simple but important points to indicate that there are still major challenges in allogeneic stem cell transplantation, despite the major improvements that we've gone through in the last decades. So here's gene therapy comes in. We know the genetic defects in SCID patients. So if the technology would allow us to correct or restore these genetic defects, then we could use the patient as his own donor, using the patient's own stem cell and correct them ex vivo and reinfuse them in the patient to restore the defective immune system. This would have several advantages. First, you always have a donor because you're your own donor. Secondly, you would, by definition, avoid the risk of graft versus host disease because you're using autologous cells. And of course, then the main remaining question is, can you do this in an effective way and in a safe way? So is the procedure an effective and safe enough alternative for allogeneic stem cell transplantation? So is gene therapy already used in SCID? Yes, it is. It is being pioneered already for 15 to 20 years. Went through a very important and impressive learning curve. And today we can conclude that for at least two types of SCID caused by the ADA gene, as you can see encircled on the left, and the common gamma chain encoding gene here encircled in the middle. For these two types of SCID, gene therapy has been proven to be effective. For ADA, it's even that well analyzed that it is now an approved clinical treatment with clinical licensing. And for the common gamma chain deficient SCID, it is being explored in quite a number of patients. And especially in the recent experiences, these results were really successful. For the Artemis variant of SCID, a clinical trial was recently initiated in North America, and our favorite gene, RAG1 gene, is the topic of this afternoon. So in summary, we can at this moment conclude that there is clearly a medical need to have an alternative treatment next to stem cell transplantation for SCID, for RAG1 SCID in particular. And the Recomb project has the focus to make this stem cell gene therapy available in a patient-centered approach to give patients a hopefully effective and safe procedure, which will allow also the patients to be treated in its own center and not having to travel to a single expert center somewhere in Europe. And here I would like to end, and I suppose Karin will take over in a few seconds.
Thank you so much, Arjan, for this very clear overview, a great way to introduce the webinar. So next up with us is Professor Karin Pike-Overzet, who will explain how gene therapy works for RAG1 SCID in more detail. Professor Pike-Overzet is working as a senior scientist in the Department of Immunology of the Leiden University Medical Center, still in the Netherlands. Professor Pike-Overzet, the screen is yours. Thank you so much.
Thank you very much, Johan, for this kind introduction. So I am an immunologist by training, and I'm very much interested in molecular work. So I will be in charge of explaining a bit more about the technical details about what is RAG-SCID and also how we develop gene therapy for this. So I'm not sure if my slides are advancing. Yes, here we go. So this is what I'll be discussing. What is RAG-SCID and also how we develop the gene therapy for this. As Arjan already mentioned, RAG1 is involved in the development of B and T cells. Mostly for B cell development, this happens in the bone marrow. From stem cells, the B cells can develop. T cells, however, can only develop in the thymus. So the stem cells first have to travel to the thymus and there undergo development and become mature T cells. And these T and B cells, they have a huge variety of different receptors. So they can recognize also a big variation of antigens or foreign objects that they might observe or encounter. So I'm going to talk to this slide because RAG1 as well as RAG2 are very important for recombination. And this recombination is needed to make all these different kinds of receptors. So on the top, you can see an example of our genome of one of these receptors. And basically, with the help of RAG1 and RAG2 and a bunch of other proteins, the little pieces can be cut and pasted together, and this is done in a more or less randomly fashion. So in the end, you get all different kinds of combinations that in the end become all different kinds of receptors, helping us fighting for in our immune system. So in SCID, of course, there's no T cells, and in RAG-SCID, there's also no B cells. When this happens, we want to find a solution for that. And this is what we intend to do with gene therapy. So we take the cells of the patient, and these cells are then enriched for being really the more pure stem cells, so the kind that will give a long-lasting reconstitution of the immune system. And these stem cells are then transduced with a lentivirus that carries the correct copy of the RAG1. And this stem cell product is then frozen. And we do this so that we can do quality control because we really want to make sure that the cell product that we attempted to make was indeed correctly made, so that we can be sure when we give the product back to the patient that this has the best possible chance of being successful. And we have shown in our Recomb project actually that we can make these products in the clean room. We have shown that 5 or 6 times that we can make a product that has sufficient RAG expression in it. And then after you infuse the stem cells, of course, you hope that the B and T cells can again develop with various different receptors and producing antibodies by the B cells. So we use lentiviruses. That sounds a bit strange, but the lentiviruses, they actually have some very good qualities that you can use for gene therapy. So it can bind and enter the cell, of course. Its viral genome is, of course, RNA. This can be reverse transcribed into viral DNA. And this DNA can insert into the genome. And after this viral DNA is transcribed, the building blocks for new viruses can be translated, and the virus can be built up and release more virus. Of course, this last bit we really don't want for the gene therapy, but the vectors that we use that were engineered about 2 decades ago, they do have some of the features that we like. So it can be a vehicle to get our transgene into the cell. it can be inserted into the genome, which is what we want because we want a lifelong cure. So when the cells start dividing, you will still have a copy of the gene in the daughter cells. And the last thing that we also want is transcription and translation of our therapeutic gene. So RAG1 will be expressed to appropriate levels. So we'll take a bit of a closer look to how these vectors look when they are inserted into the genome. You see here 2 examples of such vectors. For us, we want to have, of course, RAG1 in there in the correct version. And these vectors have so-called promoters. So on the top, you see a promoter that is rather weak. If you look at the picture on the right, you can see that there is expression of RAG in the circle, but it's not really that high. When we look at a different promoter, in this case MND, here you see that the expression of RAG1 is much higher. So we tested over the course of several years, I think about 10 different vectors. And they all had different promoter strengths, and we did this so we could find the appropriate vector to express RAG1 at the appropriate levels. So what we use for that is mouse models. So we use a RAG1 deficient mouse model, and this model has, just like the SCID patients, a deficiency in B and T cells. So for us, it is nice that this animal exactly mimics the situation in the RAG1 SCID patient. So when we test these vectors, we take the deficient stem cells of these mice and do the same procedure. So we transduce it with a RAG1 vector, and then we transplant it into these mice. And then we follow up in the blood what has happened. Do we get B and T cells back? How long does it take? And we actually saw for several vectors that it was not working at all, or that it was working only a little bit. But then the vector that I showed you in the previous picture, the one with the really strong promoter activity, that one would give sufficient B cells and T cells. And in addition, we could also get antibodies. So the B and T cells, they could work together to activate the B cells to produce antibodies, which is of course also very important for treating SCID patients. And lastly, we also could detect sufficient rearrangements. You can see on the bottom a gene therapy treated mice, and from different organs we could see that the rearrangements had taken place. And then if you look slightly above, you see these flat lines. Those are actually the non-existent rearrangements in the RAG1 deficient mouse. So we go from no rearrangements to very good rearrangements in these mouse models. And as I said, we have shown in our clinical trial build-up that we can make this product with sufficient expression to correct the disease. So I would like to stop here and acknowledge all the people that were working on this for a very long time, immunologists, pharmacists, clinicians. It was a lot of work to get this going, and it seems like we have been able to pick the right vector with the help of all these people. Thank you.
Thank you so much, Karin, for the great presentation. I see that questions are starting to be tabled under the questions tab. As we said at the beginning, we'll take them after the last presentation. So now it's time to look into the status report and prospects of Recomb. And presenting this is Professor Frank Staal, who is Professor of Molecular Stem Cell Biology, co-head of the flow cytometry core facility at Leiden University Medical Center, and guest lecturer at TU Delft and Erasmus MC. He's also the coordinator of the Recomb research program. Professor Frank Staal, over to you.
So the future of RAG1 SCID gene therapy, the future of the Recomb project, and gene therapy for autoimmune disorders are topics I like to discuss with you this afternoon. And by the way, Johan, thanks for the nice and kind introduction and the opportunity to present our work here. As already explained by both Professor Lankester and Professor Pike, but I'd like to rephrase it a little bit, gene therapy for RAG-SCID is really very simple in its beauty. It is basically taking the patient's own stem cells, the autologous stem cells, bringing them in the laboratory together with a viral vector that encodes the gene that is missing, so in this case RAG1, making sure that that gene is now expressed in the patient's stem cells and then reinfuse them into the patient. While this seems very simple, in reality, it's a very complex path for the development. And that is shown a little bit in this time frame, and this starts at 2012, but in reality, 2007 was when we really started with a different type of vector. In 2012, we started this project, and we first tested a number of vectors, as Karin indicated. We moved to the pharmaceutical phase to make sure that the vector of choice was made in a larger scale and under GMP conditions. We were very much helped in that process by a local biotech company, Batavia Biosciences. And once we had that in place, we could start our clinical phase, which also involved testing the actual protocol under the conditions that we will use with the patients. So in our clean rooms, with human stem cells, with millions and millions of them that will be used also in the patients. We started our regulatory documentation and will soon be able to include patients. And I'd like to acknowledge our colleagues in London at UCL and Great Ormond Street Hospital for their help, but also, of course, our other clinical Recomb partners and IPOPI, who have been really instrumental in bringing this forward. This will be the first stem cell-based gene therapy in the Netherlands and the first gene therapy for RAG-SCID globally. Now, what it will then involve, I'm happy to say that we have already obtained full regulatory approval in the Netherlands, and we will now spread this to the other countries participating in Recomb. The vector, as Karin already introduced, is a lentiviral vector, which contains the MND promoter driving a codon-optimized version of the RAG1 gene, and we aim to treat 10 patients for starting. We used our own stem cells, and as Professor Lankester introduced, very importantly in the Recomb project is that the stem cells will travel to our production center in Leiden from 11 clinical centers across Europe and Israel, and then we send back to the patients and to the national centers for the patient care. We will use standardized monitoring, both clinically, cellularly, and molecularly, and importantly, as part of Recomb is a full ethical and socio-economical evaluation, but also we will look at quality of life and health economics. Now, basically, the Recomb model is, I think, attractive for many of these gene therapies because there is no foreign donor needed, as with any gene therapy, which reduces the chances of graft versus host disease, as already expertly introduced by Professor Lankester, but also the treatment will be traveling. And that brings together the fact that Recomb is a collaborative consortium of researchers, clinicians, and patient advocacy. And we're very happy to make our progress available to you in this webinar. Now, as indicated by Dr. Pike, it's not so easy to develop these gene therapies. It actually takes a very long and complex path to develop the laboratory part till you have reached the situation that you know which vector to use. And then becomes also difficult to produce that vector in GMP grade at the high amounts that you will need to treat a patient. At the same time and simultaneously, the regulatory process is starting, in the end resulting in a phase 1, phase 2 clinical trial, which hopefully then will result either in a phase 3 trial or for some of these diseases immediately into clinical trial market authorization. We think market authorization is important because the product then becomes a licensed medicine and will be available to many more patients than just patients who can participate in a trial. If we look back on the challenges and our current challenges in Recomb, the preclinical path clearly is not so easy. Vector production has also been difficult, even though we were lucky that we have a local company that now has provided us with 3 batches of GMP grade vector at very high titer. But in general, this is costly, 1 to 5 million euros for a batch for a few patients, and there are still often uncertainties in timing. Then after you have obtained your vector, there is still safety testing to be done on this vector batch. That has to be done by a different company and is also quite cumbersome and expensive. And because we're dealing here with rare diseases, multinational, multi-center clinical trials are needed, which automatically make the management of such trials much more complex. Nevertheless, we still think gene therapy for these diseases is very promising. Where are we currently with Recomb? Well, we deal with certain uncertainties, as I'm sure all of you do, due to the COVID-19 pandemic. We have had some delays, and we certainly will have extra costs. What also has become apparent is that the voluntary harmonized procedure for regulatory approval is now a difficult path to follow, and that's because this path is now no longer used in, for instance, the UK and Germany. So while we thought originally that one center would be the key to obtain regulatory approval in all other countries, that way of getting regulatory approval is now no longer an option. So we'll go with national approval. We've already started with the UK and we're pretty far there, and we'll very soon go to the other countries so that hopefully within the next couple of months we will be able to start spreading it out over the EU. Another aspect is the audits of clinical centers, which can be very costly, but we have found a solution, we think, by using the LUMC auditors to go to international centers. And when we think further beyond Recomb, Recomb is aimed at RAG-SCID, but there are other mutations where also the RAG1 gene is affected. This is, for instance, in Omen syndrome and certain types of SID. For these diseases, it is very possible that the same kind of vector approach would help. And we already have some data from Milan in the Recomb consortium, Professor Anna Vida's lab, that in mouse models for Omen syndrome, lentiviral gene therapy might be of clinical benefit. What is also important to realize is that we may go for commercialization. We want to do that in a responsible way, and we have already patented this approach for RAG-SCID. When we think a little bit further about legal and regulatory challenges, I've already talked about the voluntary harmonized procedure, but there is also the issue of commercial licensing versus the academic way of doing this, an academic treatment, as we know, for the allogeneic stem cell transplantation. Sometimes for these rare diseases, commercial licensing is really difficult. It may not be possible or may even be contraindicated because of the cost. What I think is going to become very important is that we get global harmonization in rules and also in the way we conduct these trials. That requires standardization of materials, rules for stability of the product, and also ways to deal with batch differences, because invariably you're going to make new batches where you include more patients. The pricing of these gene therapy products is a topic of concern, and there are a lot of people in the field who think about this. When we look at a couple of gene therapy products, such as Trimvelis for ADA-SCID, but also Zynteglo, which are both stem cell-based gene therapies, then you see the cost of these therapies are very, very high. For Zynteglo, even around 1.6 million euros. And other gene therapy products that have reached the market, such as Luxturna for the eyes, it's a different product, is still really expensive. And this even excludes the cost for conditioning, the ICU, repeated visits, etc., etc. So it brings a discussion about accessibility versus affordability. If you want a more American model where there is free pricing versus European model where we really want to have controlled healthcare systems. Of course, these therapies, in principle, are just one-time treatments that bring cure for life. That means that there could be a way to give them a special price, but I think still it's very important to realize, are we going for value-based pricing or for cost-based pricing? And that's a discussion that is very important in the field to have. When we look into the future, stem cell-based gene therapy is possible for a number of diseases and will become more widespread. I think other types of SCID, of course, RAG-2 comes to mind, but also other immune disorders like XLA is very well possible. Thalassemia, sickle cell disease, and interesting a number of metabolic disorders can all be treated by stem cell-based gene therapy. Hyper-IgM syndrome is a different one. There you would really need controlled expression, and that comes to a topic that I don't have time to discuss today, but it's very important, about gene addition versus gene repair. What we're currently doing is gene addition, but there are ways, for instance, CRISPR, to use gene repair. However, for immune disorders to really repair a gene, this is not so easy yet. So for clinical implementation, currently gene addition is still the easiest and the first way forward. I talked about GMP pricing. We really need to address that. And we should look in the future at non-viral ways to treat the stem cells and to treat our patients, to bring the gene in a different way that is less expensive and less complex. Regulatory issues are important, and I would really hope that we get in Europe new legislation to make this less complex. So finally, a remark that I think is really important, that may be my most important slide this afternoon, that has to do with the involvement of patients in these advanced therapies. Patients, I think, should be involved in prioritizing translational research and should be involved in the grants, the grant writing and the design of the therapies. Patients can influence politicians, policymakers, and also grant funding agencies. And if that is not successful, they could even start their own charities and thereby prioritize and push the science. And I think a great example of this is what AFN Téléthon in France has done for diseases called leukodystrophies. Those are neurological diseases, but they are treated by the very same way. That means autologous hematopoietic blood-forming stem cells with lentiviral vectors. And that is a very nice way to bring forward therapies using this technology. To conclude, the combination of stem cell therapies using either gene editing or gene addition promises many curative therapies for, I think, a whole lot of severe immune disorders over the next few years. And with that, I'd like to end, not till I actually acknowledge one person, and that is Laura Garcia Perez, our very talented PhD student in the lab, who has really brought the gene therapy project on RAG1 to the next level. And she's from a very small country, Andorra, but she made big steps in bringing this gene therapy forward. So thanks to Laura, I'd like to really acknowledge you here, in addition to all the people listed. So thank you. And now I think we're very much ready to answer your questions.
Absolutely. Thank you so much, Frank, for the great finale of the presentation part of this webinar and the invaluable insights you provided on the Recomb project. And certainly bouncing on your last slide about the power of patients, we can certainly say at IPOPI that it's been not only a pleasure, but I think it's been really important to involve the patient perspective in the Recomb research program. And it's not only the patient perspective, it's really the whole input into even the governance of the research program. I think today patients bring their own expertise that can be very complementary to our expertise. And it's certainly a nice example what we have done with Recomb. I would say that some of our national member organizations are already themselves getting involved in some research programs, but certainly for those watching this webinar, either live today or the recording later, I hope this showed a great example of what the patients can also provide in the context of a research program. And I would encourage them to certainly seize the chance, if they have it, to do the same. So now, after this excellent presentation, we can see that we have quite a few questions actually in the question tab, so time has come to answer them. If you still have a question in mind, there is still time to ask it. I don't promise we will go through all of them for the sake of time, but we'll certainly do our best. So perhaps starting with our first presenter, Arjan, there was a question about the possible risk for the patient. So I think someone talked about the risk of leukemia in the past and the safety. What would you respond to that?
Yeah, thanks, Johan. I think that is probably one of the most important questions, because of course we all know, or maybe not everybody knows, but there is a history of problems in the early days when gene therapy was explored in clinical trials for other types of SCID. And I think the most important lessons learned from those early trials was that other types of vectors, without going too much into the technical details, but other types of vectors were needed to obtain the same efficacy, because the treatment was not ineffective, it was just associated with side effects. And using newer generations of vectors, the so-called lentiviral vectors in particular, as Karin so nicely explained them, they are, as far as we know right now, and the follow-up is already a considerable number of years, and the number of patients treated with these new generation vectors is already substantial, not only for SCID, but also for other diseases. They seem to have a very favorable safety profile, and no, let's say, malignant events have occurred in patients treated with this newest versions of the clinical applicable vectors.
And Arjan, maybe building on that, what do you think would be the possible advantage for the patient to go for gene therapy instead of HSCT?
I mean, if you look at it from a clinical perspective, I think the main hurdle of allogeneic stem cell transplantation is not so much the efficacy, but it are the side effects associated with allogeneic stem cell transplantation. The most profound one is a graft versus host disease, which in its severe forms can be lethal. And that is something that you would like to avoid, but not just graft versus host disease, also some other, I would call semi-graft versus host-like phenomena related with an allogeneic inflammatory response that may occur, especially in patients that are vulnerable or affected with infectious complications, may be a real hurdle. So if stem cell gene therapy is effective, and we are very confident it is, then it should be at least as effective as allogeneic stem cell transplantation and avoid the typical risks associated with allogeneic stem cell transplantation.
Thank you very much, Arjan. That was a very clear response. Karin, maybe some more technical questions in the question tab for you, perhaps starting with this one. For how long can the cell product be kept frozen before infusion into the patient, and how stable is it?
Yeah, thank you very much for that question. Of course, when we want to treat a patient, we want to keep this period of time that the cell product is frozen as short as possible. But we think we can do the quality control in about 3 to maybe 4 weeks. But of course, we have tested longer time periods that we can keep the product frozen. And at 3 to 4 months, the product is very stable. We have really good viability and still really good transgene, so RAG1 expression. And even we did some stability tests up to 8 or 9 months for the test products that we made. And even then we saw good viability and good transgene expression.
Excellent. Thank you so much. There's a couple of other questions. I'll go with this one. You showed that the MND promoter gives higher RAG1 expression. Is more always better, or can there be a situation where you get an adverse effect from too much expression?
Thank you for that question as well. So For RAG1, we tried a lot of different vectors, and in most cases, the promoter was not strong enough, so we ended up with one of the strongest promoters to use for the clinical trial. In parallel, we're also developing gene therapy for RAG2, and we were going along the same path, like, okay, let's use the stronger promoters to express RAG2, but then we didn't get as much success as we thought we would get. And actually, when we tested some weaker promoters, we actually saw that in this instance for RAG2, the lower dose was actually better. So this probably has to do with the function of the RAG1 and RAG2 in the cells being a little bit different, and high RAG2 expression apparently is not so such a good situation for developing B and T cells.
Thanks so much. Maybe a last one. How is RAG1 expression measured in the cells?
Yeah, so often when people measure things on stem cells and cell products, this is measured on the protein level, and usually it's something that you can do on the cell surface, or maybe like an activity assay using enzyme activity. However, we don't have such assays for RAG1 and RAG2. The readout we have for functionality is an in vivo experiment that will last several months. So we measure RAG1 on the short term on RNA level. So we either take our culture and we measure with a PCR method the RNA, the expression level. But you also saw the pictures, and those pictures are made on a flow cytometry. So we can see on a cell-by-cell basis what is the RAG expression. And this is a sort of an RNA probe that is connected with fluorescent fluorophores. So we can see on a per-cell basis what is the RAG1 expression.
Thanks so much, Karin. Maybe now switching to Frank a little bit. This question, which is really two in one, and the second part of the question, I think is similar to a couple of other questions that have been asked. Do you have any idea about when the Recomb will be able to start with the first patient? That's the first part of the question. And then the second part is about the huge and promising step for science. What other types of SCID or even PIDs could potentially be candidates for gene therapy approach?
Yeah, very good questions. I think we will very soon be able to have patients, if they are in the Netherlands or surrounding there or could travel to the Netherlands. I hope that the other countries, and it depends a little bit on how the national regulatory process will go, will also follow within the next few months. We are pretty far with the UK. So I think then we could open up the trial, but I would anticipate that sometime this year we could have the first patients treated. It also, of course, depends whether they fit the inclusion criteria. As Professor Lankester explained, we only for now will start with patients that don't have a matched donor. So if there's a matched donor available, we will not treat them in this trial. That is because of reasons of safety, that we'll make sure first that this gene therapy is safe and effective before we move to patients that now have a very good alternative. But I think we will probably find those patients and they will start getting treated in this trial. Then if we move forward to other indications, I like to make a distinguishment between those genes and those diseases where we can use the gene addition approach versus those diseases that require regulated expression. So RAG2, we now have a good vector. It was a little bit more complicated, as Dr. Karin Pike just explained. There it was not the more the better. We really have to have a certain level, but you can control that. With other diseases, you need maybe specific expression in a certain lineage, which we know is the case for XLA, which we are working on with gene therapy as well. But for some other diseases, say IL-7 deficiency, IL-7 receptor deficiency, the receptor, or the hyper IgM syndrome, it's clear that you need regulated expression, and that's a little bit more difficult. I think it will get there because there's so much research going on worldwide that will be available. That will take a little bit longer. So I think for many diseases, a good hope. I noticed there was a question about CVID, which is a complex disease because it is actually multiple genes are affected often, or we don't know which gene it exactly is. And then, of course, gene therapy is more difficult. I think for CVID, we'll need more research to know what is the underlying gene defect, because that's a precondition before you can actually start with gene therapy. You need to be absolutely sure that the gene that you're going to give in a vector is actually the gene causing the disease.
Thank you so much, Frank, for that. Maybe a couple of the last questions, and feel free, one of you, to answer them. There's one which is obviously an important question about the product traveling rather than the patients. And, what are the hurdles in that equation, and what could we do to make it happen? Who would like to take that one?
It's probably more related to your experience, but travel.
I think the aspect of traveling of the patients or the cells is, I think, a very fundamental issue, because I'd just like to give briefly an example that which we encountered more than a year ago, where a patient in our unit met the criteria to join a gene therapy trial for one of the other SCID genes. But because of, let's say, social reasons, the family decided not to participate because it implicated that they had to travel abroad and stay there for several months. So it was not so much their trust in the therapy, but it was simply for social reasons. And that is something that I think shouldn't be underestimated. The impact of traveling with a newborn baby. I mean, try to imagine it, a 3-month-old baby, you leave your house, you leave your family, you go abroad, stay there for several months. Even in a period without Corona, that is more than a challenge. So to serve the patient, I think setting up a structure like Recomb aims for, to serve the patients, to keep them in their natural habitat, in their own expert center in their country, which is part of a larger consortium, which brings the treatment to the patient and not the patient to the doctor, is, I think, something that we should really aim for. That is the future, and this is, I think, an important step in this process.
I would fully agree. Thank you, Arjan, for that. Maybe the last question, 3 Dutch speakers, we know, and we've heard the great news that the Netherlands were starting to screen for SCID as of 1st of January of this year, so a very new development. How would you describe the importance of newborn screening for SCID in speeding up the access to treatment and making sure we don't miss these babies? How important is that in your eyes? And again, whoever wants to reply this last question.
Yeah, I think this is an important issue as part of the committee to advise the government to bring SCID into this neonatal screening program, and it's being done all over the place in Europe now. In the United States, they've had it for many years. So I think it's very important, because we know if we recognize the patients early, the earlier we can start with proper treatment, whether it's a stem cell transplant or a gene therapy, the better. It's also, and Arjan can say more about it, and he should, I think, it's also a little bit strange compared to what used to be the case. It used to be the case that babies got ill and they were seen as a patient, and you go to the pediatrician and then the diagnosis SCID would be there. Now, a healthy baby and parents who are very happy with their healthy baby get some unfortunate news out of the neonatal screening program. So that is a very different type of psychology, and I think very difficult for the pediatricians to convey that message. But I'm sure that Arjan can say some good things about this, how to deal with that.
Yeah, I fully agree, Frank. And I think the newborn screening is something that should be used. What I didn't put in my presentation, but could have done so, is to show the impact of starting the transplant, but the same would hold true for gene therapy in a healthy SCID patient, meaning without infectious complications or affected. And it really has a difference of 15 to 20% on overall survival. So only from that perspective, early recognition, early diagnosis is seems to be an important aspect of the overall treatment and outcome.
Well, thank you so much to all of you indeed. It's a great way to finish the discussion. I think the fact that we still have most people with us, although we are a little bit over time, is a great testimony to the interest in the topic we discussed today. So we have now reached the end of this very first Recomb webinar, and on behalf of IPOPI and Recomb, I would like to say a warm thank you again to all our speakers for their excellent presentations and for all the answers, very thoughtful answers indeed you provided in the discussion. And just before we finish, if you have a minute to spare, we encourage you to share your feedback on the webinar by pressing the link which you see appearing on your screen now. Don't worry, once we shut down the webinar, if you've clicked on the link, you have plenty of time to fill in and complete this feedback form. And that will be very helpful to us to get your perspective on how helpful you thought the webinar was, whether we should think of doing more of these sort of educational initiatives and so forth. So please note that you will be able to see the recording on the Recomb website within a couple of days. So if you want to, if you've missed part of the webinar, or if some of your colleagues would like to watch it, you can encourage them to visit the website and to share it as much as you can with whoever you think will benefit from it. So thank you again, everyone, so much for attending the webinar. Stay safe, take care, and see you next time.